I'm Emma Fryer, and I'm one of NISHU's master's students. Most of our lab works on geoecology, so how geology shapes plant diversity. I'm working with this group of about a dozen annual plants from the San Joaquin Desert that are endemic to verdic clay soils. So those are extremely high clay content soils that have very high shrink swells. So when they get wet, they expand a lot, and when they dry, they contract even more, which creates the characteristic cracking that they're known for. You can kind of see here the soils at both of my field sites, akin to a creek in Fresno County and Carrizo Plain National Monument, are very heterogeneous, and they vary across a range, though they tend to be more on the extreme side. They range from non-sodic to extremely sodic. Some are high in selenium, some in gypsum, and in general make for a pretty harsh substrate for plants to make a living on. So all of my study species, which occur in very distinct patches, seem to be adapted to really extraordinarily difficult to live on soils. Most of the year, the San Joaquin Desert is very dry, very hot, and very brown. However, every few years, after a very good rain year, we see what we now refer to as superblooms, and which now are all over social media. The heterogeneous soils at my field sites translate to very distinct patchworks of color across the landscape that are very vibrant and are basically single species stands of my study species. My project is to figure out how soil chemistry, soil texture, and competition from the invasive grass Bromus madridensis are all coming together to influence the community assembly of this plant community and to create those very dramatic patterns that we've all seen. So while I thought I was going to be doing it in Cal Poly's greenhouse, I'm instead in a friend's backyard doing my study here in Fresno. I have several studies running at once right now. I have 12 study species, and what I am doing here is I have them planted across 3 different soils that represent a gradient of stress, and I am doing a duplicate study mixed in with it where I have added Bromus madridensis seeds as competition. And over here I'm addressing soil texture. It's a 2-part study. I have a group of 5 of my study species and then 5 congener species that are native to contrasting soil textures, and I have reciprocally transplanted both of those species across both soils and done the same in these devices, which are called rhizotrons, that allow me to look at the roots. And through the combination of soil texture, soil chemistry, and the role of competition, I'm hoping to figure out not only what is going on in this community, but also the nature of these plants' adaptations and how verdic clay endemism works.